So in the last class, we have almost completed the structure of, okay. In the last class, we have seen the composition of the cell membrane. In the cell membrane, what we have seen that the major important components that constitute a cell membranes are lipids, proteins and carbohydrates. Now, lipids constitute the major part because it is a lipid bilayer, and specifically the lipid that is present is specifically the lipids that are present in the cell membranes are phospholipids. These phospholipids have a glycerol head will be there, that is hydrophilic in nature, and then they'll be having the fatty acids which are hydrophobic in nature. So this is arranged in this order. Now the proteins are embedded into the proteins are embedded into the cell membrane. So these proteins, the form in which they are present on the cell membrane, they are divided into two types: the integral proteins and peripheral proteins. The integral proteins are the ones that pass through and through the cell membranes. The ones that pass through and through the cell membrane are called as integral proteins in that way. And peripheral proteins are the ones that are present on the outer surface. So they could be present towards the cytosol, or they could be present outside the membrane, but they are present only on the surface. They do not pass through the lipid layer. Now, one more thing that we usually see are the carbohydrates. These carbohydrates are usually oligosaccharides. That means each one is one monomer. So up to few monomers, up to 10 monomers, they are usually present. So they are called as oligosaccharides. And these carbohydrates are usually oligosaccharides. They are associated with the lipid part of they are associated with the lipid part. Now, along with that, one more type of the lipid that is present, which is called as a cholesterol. Cholesterol is another type of lipid that is present in the cell membrane. So, totally, if we consider the lipid part are two types, cholesterol and phospholipids. The phospholipids constitute the major part of the cell membrane, major part when compared to the proteins and also with the carbohydrate. Then the proteins that are integral part, they can be integral proteins or they can be peripheral protein. Then cholesterol to some extent, and then carbohydrate, they are present. So these carbohydrates are called as a glycocalyx. They mainly help in, we have also seen that what are the functions of individual proteins. The membrane is mainly formed by the phospholipids. The integral proteins are the peripheral proteins. They help as transporters. They help in signal transduction. They help in giving support to the cell membrane and all that. The cholesterol that is present, it will help in maintaining the fluidity of as I told you, this is a fluid mosaic model. Mosaic, we understood mosaic because if you look at this overview, you can see some white parts, some blue parts, some green parts, some yellow part, like that. So it is looking like a mosaic. Now, the fluidity is determined mainly by the fatty acids that are present here and the cholesterol that is present. These two will determine the fluidity of the cell membrane. So this is a bit, and now and regarding the functions of carbohydrates, the carbohydrates, they mainly help in cell to cell connection. As we know, these are negatively charged. So because these are negatively charged, they do not allow two cells to come together. Then how will a cell communicate with another cell? This is done by the glycocalyx, glycolipids. That is a carbohydrate combined with the lipids. They are called as glycolipids. So these help in maintaining the cell-to-cell interactions. Now, we learn a little bit about what is this fluid mosaic model. Fluid means the one that always changes, you call it as a fluid. Mosaic means beautiful arrangement of different colors. That is mosaic. Now, mosaic we understood because when you look from the surface, you see multiple colors because it has got multiple components. Now, why do we call it as a fluid? Now, this was given by Singer and Nicholson. According to this model, membrane proteins are like icebergs floating in the sea of phospholipid molecules. That is what we have seen in the previous diagram. You see just the upper part of the protein, but when you go deep into that, you can see the membrane proteins, they are crossing through and through the phospholipid layer. Phospholipids undergo rapid. Now we'll talk why you call it as a fluid. Phospholipids also undergo rapid redistribution in the plane of the membrane. Due to the fluidity of the membrane, the appearance of the proteins seen by electron microscope membrane structure is called as fluid mosaic structure. Now, the fatty acyl chains in the interior of the membrane form fluid, that is a hydrophobic region. The integral proteins float in this sea of lipids. Both lipids and proteins are free to move laterally in the bilayer of the phospholipid, but the movement is either from one phase of the bilayer to the other phase. That is flip-flop movement is restricted. It is only through one phase. Now I'll explain it further. So these fatty acyl chains that are present, as I already told you, the fatty acyl is the one that is mainly responsible for maintaining the fluidity of the membrane. Now too much of the movement, because they are hydrophobic in nature, they are very dynamic. They don't want to be associated with any of the aqueous media. Now the presence of these integral proteins, they prevent too much of movement of the fatty acids. Now if the lipid layer is moving too much, obviously the stability of the cell will be lost. So too much of movement is prevented by the integral proteins. Now what do the carbohydrate moieties do? The carbohydrate moieties attach to some proteins and lipids of the plasma membrane, which are exposed on the extracellular side. So, in that, the movement of the lipids will be like this. The movement of this will be on the lateral. They do not flip-flop. If they flip-flop like that, everything will turn around. So, the movement will be in the fluid mosaic model. In the fluidity, the movement of the fatty acids will be in this movement, lateral movement only. There won't be any flip-flop movement. Molecules rarely flip transversely. This is very rare across the membrane because hydrophilic part would have to cross the hydrophobic core. Because if they are flip-flopping like that, these are hydrophobic. They have to come into touch of the hydrophilic aqueous part. Here and there aqueous part is there. They have to come into touch with that. So never the glycerol, and this part doesn't move. Only the fatty acids that are present inside, they do the movement. The phase changes and the fluidity of the membrane is largely dependent upon two factors. It is dependent mainly upon the lipid composition and the temperature. It is mainly dependent upon the two. Now, we'll see the lipid composition. What is the lipid composition that is determined by it? Cholesterol and saturated and unstructured fatty acids, it is saturated and unsaturated fatty acids in the phospholipids. These determine the fluidity of that. Cholesterol will try to make it stable. Presence of saturated and unsaturated fatty acids make it more fluidy in nature. So the fluidity is mainly dependent upon the two factors, lipid composition and the temperature. So lipid composition, cholesterol tries to make it more stable, whereas saturated and unsaturated fatty acids make it a little bit more fluidy. Now, temperature. The temperature is called as Tm, is a temperature where the cell will be existing normally. If the temperature increases, the fluidity will increase. If the temperature decreases, the fluidity will be maintained like that. And that composition is mainly affected by the cholesterol. So, as the temperature increases, the cholesterol is much more stable and it prevents the flipping of the, not flipping, but it will prevent the movement of the fatty acids. So the fluidity is determined by lipid composition and also by the temperature. Next we'll see about the, so this is somewhat about the cell membrane and all that. Now, one more thing that we have to cover in the cell membrane are the transporters. What are the transporters that are present in the cell membrane and all that. Now, before going into the transporters, because we have already covered all the cell organelles we have seen, now we'll see the marker enzymes. What do you mean by a marker? Marker, as I told you, marker is a one that will help you to identify one particular. As I told you, you have almost 10 things you have arranged, but accidentally you have to put all of them in one cover. So you have put your pens in one cover, or all the erasers in one cover, color pencils in one cover, like that. But the colors are looking, the colors of the cover are same. You couldn't get different colors. So what you do? The next step to recognize what is what, you'll put, you'll write something on the cover. These are pens, these are color pens, these are erasers, like that. You mark each cover. In that way, each cell organelle has got a specific marker. Now, when that enzyme or that marker increases, the amount of that marker, if it increases, you will identify that, okay, there is a problem with this particular cell organelle. So that you call it as a marker. A marker or a chemical is one that is almost exclusively confined to one particular organelle. The marker thus can serve to indicate the presence or absence of the organelle in any particular fraction in which it is contained. So, the marker will help you to identify whether that particular organelle is present or if it is absent, or if there is some problem that is associated with that particular organelle. In that way, each cell organelle has got a specific marker. So this is a thing that you have to remember, the markers. Now we'll see why this marker is specific for that particular organelle. Most important is the mitochondria, because it is the currency generating cell organelle. So now if there is any problem with the, and its major functions are, it is involved in citric acid cycle and oxidative phosphorylation. Now one more criteria in which, by which we pick up it as a marker. Mitochondria has got so many enzymes because it is a center point for so many metabolical processes. Then why did we select only glutamic dehydrogenase as its marker? We have selected this as its marker because the reaction that involves this, the reaction that utilizes this enzyme, that reaction occurs only in mitochondria. Nowhere that reaction occurs. That means that this enzyme is exclusively present in mitochondria. It is present nowhere else. So that is the reason we have picked up this enzyme, and this will give you, and in that way you can see that citric acid also takes place in mitochondria. Then why can't we pick up the number? No, there is some functioning relationship of mitochondria with this. Some functional relationship. For that reason, the glutamate dehydrogenase is the one that is selected as marker for mitochondria. Now, for endoplasmic reticulum, glucose 6-phosphatase, because this enzyme is present only in endoplasmic reticulum. This glucose 6-phosphatase enzyme is utilized to convert glucose 6-phosphate to glucose, to normal glucose, without any substitutions attached to it. So this enzyme is present only in endoplasmic reticulum. That's why this glucose 6-phosphatase is used as a marker for endoplasmic reticulum. Now, what are the major functions of endoplasmic reticulum, which we have already covered? Its membrane bound ribosomes are a major site for, we know, if it is rough endoplasmic reticulum, it is a site for translation, and if it is smooth endoplasmic reticulum, it is required for synthesis of various lipids. It is very much required for the metabolism of drugs. Nucleus, the marker for nucleus is DNA, because DNA is present only in the nucleus. Now just to refresh, what are the functions of the nucleus? It is a site of chromosome, where chromosomes are present. This is a place where replication occurs, where transcription occurs. Every control and everything will take place in the nucleus. Next, ribosomes. Ribosomes are characterized by presence of RNA, ribosomal RNA. And what is the function of the ribosomes? These are sites of protein synthesis. Lysosomes. The main function of lysosomes is that they do hydrolysis. They have got various hydrolytic enzymes, and these act as a scavengers of the cell. So the lysosomes, the best marker for lysosome is acid phosphatase. Now if acid phosphatase levels are drastically increasing, it means that lysosomal activity is increased, and the very high activity of the lysosome is also not good, because if it does more and more scavenging, it will eat up the normal cells also. So the marker for lysosomal function is acid phosphatase. Next, plasma membrane. Plasma membrane is sodium potassium ATPase. So the sodium potassium, in the previous class also I told you that sodium and potassium, a dynamic relationship is maintained between sodium and potassium. For every 3 sodium enters, for every 3 sodium entering, 2 potassiums are expelled out of the cell membrane. So sodium will enter in and potassium will go out of the cell membrane. So, and this is specifically organ is specific to that of the plasma membrane. So, this is as a marker. One more marker is 5' nucleotidase is another marker for plasma membrane. Next, for Golgi apparatus. Galactosyl transferase is a marker for Golgi apparatus, because this transfer reactions, the Golgi apparatus is mainly responsible for tailoring. You can consider it as tailoring. The raw material that has come out from the endoplasmic reticulum, that material will enter into the Golgi apparatus, and it is basically tailored in the Golgi apparatus. So galactosyl transferase is a marker for Golgi apparatus. Next is peroxisomes. The peroxisomes are the ones where all the oxidation process will take place. Any bacteria enters that will get indulged with the peroxisomes, and it is completely catabolized. So catalase is an enzyme, and uric acid oxidase. These two are the markers for peroxisomes. For cytosol, lactate dehydrogenase. Lactate dehydrogenase in a cells where nucleus is not there, all the other cell organelles are not there. Cytosol will provide nutrition to, like if you take the example of RBC. RBC has only cytosol. It doesn't have mitochondria, it doesn't have nucleus, it is completely deficient of all the cell organelles except the cytosol. Now what is providing nutrition to that is the glycolysis is the one that is providing nutrition to the glycolysis process provides nutrition to the RBC. So the lactate dehydrogenase is utilized. This lactate dehydrogenase is the one that is, the levels are very high in the cytosol. So they are used as markers for the cytosol. So these are all the membranes. So we have seen the markers for mitochondria, that is glutamate dehydrogenase, endoplasmic reticulum, glucose 6-phosphatase, nucleus, DNA, ribosomes, RNA, lysosomes, acid phosphatase, plasma membrane, sodium potassium ATPase and 5' nucleotidase, then Golgi apparatus, galactosyl transferase, peroxisomes, catalases and uric acid oxidase, cytosol, lactate dehydrogenase. Now, how did we come to know that each cell organelle? Now, how did we come to know the functions of mitochondria and all those things? For that, we need to do subcellular fractionation. The subcellular fractionation is done to study the function of any organelle. It is necessary to isolate it in relatively pure form. The usual process by which this is achieved is called subcellular fractionation. Now, the subcellular fractionation occurs by 3 pathways. One is extraction of the tissue, homogenization of the tissues, where the cells will come out, all the cell organelles will come out, that is called as homogenization. Then centrifugation. After centrifugation, the cell organelles will settle depending upon their mass and their density and all that. By a centrifugation, they do settle. So, subcellular fractionation is a process to study the function of any organelle, it is necessary to remove it in a form. So that form, how do you do the methods? It's called as subcellular fractionation. It is done by extraction, homogenization and centrifugation. First, extraction. From the cell, you have to extract from the cell you have to, from the tissue you have to extract the cell. So extraction is the first and foremost step in isolating a specific cell organelle. Most procedures of isolation of the organelles take place at 0 to 4 degrees centigrade. A common solution used for extraction consists of sucrose that is dissolved in tris HCl buffer, which contains potassium and magnesium at physiological concentrations. So these are the common things that are present during the extraction process. The common solution for extraction of organelles consists of sucrose adjusted to pH of 7.4 by tris HCl, and buffer of potassium and magnesium are present. When the tissue is exposed to this type of chemical, the cell membrane, everything will break and cell organelles will try to come out of the cell. Sometimes organic solvents can also be used if we want to extract lipids and nucleic acids. Organic solvents can also be used, but now we are concerned with the cell. So this buffer is usually used, and it is usually done at 0 to 4 degrees centigrade. So the three processes, as I told you, it involves the extraction, then homogenization, then filtration, then after that centrifugation. So the three methods are tissue extraction, homogenization, filtration, then centrifugation. So by subjecting the tissue to the buffer, you are removing the cells, all the dead substances, everything. Then after that, it is sent to a homogenizer where it is where a stirrer will be there. That stirrer will be continuously rubbing those tissues, mashing the tissues to a very greater extent. So it will completely mash the tissues and make it it will make it into a sort of puree thing. So it is completely broken down. It is completely mashed completely, so and it is made in the form of a puree. Now this part is filtered, and after filtering, over the filter you will have whole cells, connective tissues, what are not required, they are present above the filter. Only the cell organelles are filtered from the filter, and they will come down. Like they are filtered, all the things will remain above the filter, only the cells will come into the filter. Now cell organelles, and mentioned to you that it is not the cell, it is a cell organelles that will come to the filter. Now in the next step, centrifugation has to happen. Why centrifugation is important? Because the size, shape and the weight of the each cell organelle is completely different, and the centrifugation runs under the principle of the weight and mass. So more the density, the particles will settle down, lesser density particles will always move up. Now, when you do the centrifugation process like this, what will happen first? The nucleus and the cytoskeleton will come to the bottom, and this is done when the rotation is up to 600 for 10 minutes. If you do the rotation, what will happen? All the nucleus and cytoskeleton will settle down. In the next step, what you do, you will remove these supernatant and you will place into another test tube. So below the segment that is formed will have nucleus and cytosol, and in the second step you will remove the supernatant and you will use the supernatant. Once again you centrifuge it, and this time centrifugation is done at 15,000 rpm. For 15 minutes you do it 15,000 rpm. Now when it is done at that high frequent level, what will happen? The mitochondria, lysosomes and peroxisomes will settle down. These will be settling down. Now in the next step, what will happen? When you remove, and now you will be removing the supernatant, you will be removing the supernatant and will place it in the another tube, and next once again you subject it to the centrifugation. And now in this centrifugation you are subjecting it to almost 1 lakh times 1 lakh rpm for about 60 minutes. Then with that type of centrifugation, what will happen? The plasma membrane, endoplasmic reticulum fragments, small vesicles, microsomal fractions, all these will settle down. Then after that, you take the soup and you once again subject it to the centrifugation, and then what comes down are the ribosomes or any virus particles that are present, any macro particles, all these things will get settled at the last stage of centrifugation. Now after that, you get a clear fluid. That clear fluid is nothing but the cytosol. So the process of isolation of cell organelles involves three steps. One is extraction, in which buffer is used and the tissue is extracted from that. Then using homogenization, in the homogenization procedure, the tissue is broken down or it is mashed to form a fine puree. It is completely mashed. Then after that, this homogeneous is filtered, and when it is filtered, the filtrated part will have all the cell organelles, and the things that are left over the filter are whole tissues, connective tissues that cannot pass through the filter. The first step of centrifugation, the nucleus and cytoskeleton will get sedimented, and in the second phase, mitochondria, lysosomes and peroxisomes will get sedimented. In third phase, plasma membranes, endoplasmic reticulum, microsomes, all these things will settle down, and in the last one, the ribosomes will and in the last one, the ribosomes, viruses, and the macromolecules will form a pellet-like thing, and the superficial clear one, what you have is a cytosol. So this is a process by which cell organelles are isolated. Now, each time when the cell organelles are isolated, each cell organelle was being studied detailedly to know its structure, to know its function, and to know its markers. Everything is done so precisely by doing this method. So this is about the isolation. Now, I want to just concentrate on one or two diseases that are associated with the cell, because if I talk too much of the diseases now, you won't be having a clear idea about the enzymes, about the disorders and everything. Once we cover each and every part of the metabolism, again we'll come across these enzymes and these disorders. But specifically to tell you, as I told you, everything has to end clinically. That is the purpose of teaching biochemistry. So to end this topic clinically, I want to tell some disorders associated with that. So this eye cell disorder is usually given in cell only. When we are doing introduction of the cell, this eye cell disorder is given this only. Now this is a disorder that is associated with the defect in the Golgi apparatus. What will happen is, as I told you, Golgi apparatus is tailor in our cells. It is like a tailor. So what will it do? This will transfer. It will be transferring N-acetyl glucosamine is transferred. N-acetyl glucosamine 1-phosphotransferase. So this N-acetyl glucosamine to this N-acetyl glucosamine, the phosphate is being transferred. So what is happening here? Deficiency of glucose N-acetyl phosphotransferase enzyme, which phosphorylates mannose residues to mannose 6-phosphate. So these mannose residues are converted to mannose 6-phosphate. Mannose 6-phosphate in the presence of enzyme glucuronil N-acetyl N-acetyl glucosamine. So to this mannose, N-acetyl glucosamine phosphate is attached. N-acetyl glucosamine phosphate is attached first. So this reaction occurs in two steps. First, primarily this enzyme brings about, the purpose of this enzyme is that the mannose that is present in a particular protein. Mannose is a carbohydrate like glucose, fructose you have. In that way, mannose is also one of the carbohydrate that has to get phosphorylated. It takes place in two steps. One step is N-acetyl glucosamine phosphate. First, that gets attached to the mannose. Now, in the next step, the N-acetyl glucosamine. So, this part is being removed and you'll be getting just the mannose phosphate. Now, defect comes when there is deficiency of N-acetyl glucosamine phosphate. So what will happen when this enzyme is defective? Defective cell targeting of the lysosomal hydrolysis leads to numerous enzymes being secreted out of the cell and accumulation of their substrate mucopolysaccharides outside the cell. So what will happen? These are the ones that are that are involved in the lysosomal targeting. So these are the ones that are involved in the lysosomal hydrolysis. So what will happen? The things that have to be hydrolyzed in the lysosomes do not take place, and mainly the things that are hydrolyzed by the lysosomes are mucopolysaccharides. So these mucopolysaccharides are not being catabolized. As a result of that, they get accumulated in the cell. So that you call it as eyesight disease. I wanted to put the picture of the eyesight disease. So there will be short, they will be fluffy and all those things. The child will look completely different because it is the skin or the dermal thing. The mucopolysaccharide is mainly related to the connective tissue. So there is no proper formation of the, no proper catabolism of the connective tissue. So the cells and everything will be abnormally, and the mucous membrane, all these things will be abnormal in this situation. So this is called as eyesight disease. Next, some of the deficiency of any of the enzymes of the lysosomes are affected, that will lead to the disorder called as mucopolysaccharosis will be resulting. Now, the disorders that are associated with the peroxisomes. The peroxisome, the Zellweger syndrome is a syndrome where there is complete absence of peroxisomes. That individual will not have any peroxisomes in their cell. That is the main purpose. Now, as I told you, peroxisomes are important for the defense mechanism. At the same time, they also help in metabolism of very long chain fatty acids. The very long chain fatty acids are very long. That is why they are very long chain fatty acids. They are not normally catabolized. So they have to be broken down into smaller chain fatty acids. Then they can be catabolized by other organelles. So first what will happen? The very long chain fatty acids will enter into the peroxisomes, where they are broken down randomly into different pieces. Now the smaller pieces are metabolized by the mitochondria. Now in Refsum disease, the enzymes that are required for this breakdown are completely absent. So that is a Refsum disease, where the enzyme is called as phytate oxidase is the enzyme that will be breaking down very chain fatty acids. So that is completely absent. As a result of that, these very long chain fatty acids will get accumulated in the peroxisomes. Now Zellweger syndrome is a condition where there is complete absence of the peroxisomes. In that way, we come across various disorders that are associated with the cell membrane. Now, so as I told you, till now we have seen the cell, the functioning of what are the cell organelles that are present, the functions of each of the cell organelle. Then we have talked a little bit detailedly about the membrane, what is the composition of the membrane, and why the membrane is called as fluid mosaic model. Why and how the flipping and everything. Flipping doesn't happen, only lateral movement is common in the fluid mosaic model. And we have seen the function of individual components. What is the function of phospholipids in the membrane? What is the function of cholesterol? What is the function of proteins? And how the proteins are arranged? They are arranged as integral proteins, or they can be associated as surface proteins. And then we have seen little about the carbohydrates that are associated with the membranes. Then we have also seen that other than phospholipids, cholesterol is another lipid that is associated with the cell membrane. Then we have seen how the cell organelles are being extracted. This is done by three procedures. One is by extraction, second is homogenation, where puree is made. Then after filtering, that filtrate is subjected to various grades of centrifugation so that the components are being removed. So that part we have covered, and we have seen few metabolic disorders that are associated with the cell. Now we'll understand. So that is about the cell we have understood. Now we'll try to learn that how the things that are present in the cell are going out, and how the things that are present outside the cell are getting in, are they are coming in. So what are the ones that are controlling it? And from the past few classes, what we have seen, we have learned that the proteins are the major ones that are responsible for membrane transport. The proteins are the major ones. Now, this membrane transport is divided into two types: active transport and passive transport. It is divided into active transport, which utilizes energy; passive transport, which doesn't utilize energy. In passive transport further, they are further divided into two, simple diffusion and facilitated diffusion. It is divided into two types. One is simple diffusion, another one is facilitated diffusion. In simple diffusion, it is just like normal diffusion where the gases will enter, just diffuse through the cell membrane and enter into the cell, like oxygen, carbon dioxide, any of the gases, they just diffuse through the cell membrane. No specific mechanism is required for that. Next is facilitated diffusion. In facilitated diffusion, to move from one place to another place, they require some transporter is required. And being a passive transport, it doesn't require any energy, but it requires a transporter. Now, this facilitated diffusion, so what is a transporter? If it is a channel transporter, you call it as a channel transporter. And if it is a carrier is required, it is called as a carrier mediated facilitated diffusion. So either it would be a channel, so protein will be present and it will have a center channel through which the electrolytes will come out and come in, or it can be a carrier that will catch hold of the substance and it will take it into the cell. Next is active transport. The active transport are of three types. It is primary active transport, secondary active transport and vesicular transport. So we have seen that passive transport is simple and facilitated. In facilitated means it requires some help to do the diffusion. So this facilitated one is further divided into channel and carrier proteins. Next is active transport. In active transport, there is a primary active transport, secondary active transport and vesicular transport. Primary and active transport means it will be utilizing ATP. Energy is required in this process and we call it as an active transport. So, the active transport, primary and secondary, we'll see what are they. First, we'll see the classification of the vesicular transport. Vesicular transport means it will be forming the vesicles, and these vesicles are the ones that are exited out. They can enter in or they can be exited out. So they form a vesicles. Like normal substances, they do not enter as such. A vesicle is formed and through the vesicle, the programming will take place. Exocytosis. So a vesicle is formed. It will come to the cell membrane. It will merge with the cell membrane and the contents that are present inside the vesicles are thrown out of it. Now endocytosis, it is a process where the substances are engulfed and they are brought into the cytosol. So that is exocytosis and endocytosis. Exocytosis, a vesicle is formed and that pushes out of the substances of the cell. Endocytosis is a process where the cells, endocytosis process where these vesicles are formed and they are absorbed into the cell. Like we see the examples here. So this is endocytosis. Now endocytosis is commonly present for LDL receptors. Now these LDL receptors, normally you have a clarithin coated bit will be there, and over the clarithin coated bit the LDL particle will get attached. Now then after the second step, the LDL will get attached and it is engulfed into the cell. Now once it is engulfed into the cell, these will fuse with the lysosomes, and when they fuse with the lysosomes, this other part is removed and the LDL will enter into the cytosol. So that is example of endocytosis. So what will happen? It will have some bits that are normal in nature. When any substance that comes in contact with that, it is engulfed like this and it will form a vesicle. Now the vesicle is uncoated, it is fused with the endosome, and then after that what will happen? The recycling of the, and whatever the receptor that is being damaged, it is recycled and it will once again enter onto the cell membrane and then it will form a transport vesicle. Now, this will fuse with the lysosomes, it is digested and degradation, all this process will take place. That is endocytosis. Now, in phagocytosis, what will happen? And here, it has got the receptors. When it sits onto it, the receptors will form a vesicle like that. The difference between this and phagocytosis is that in phagocytosis, the microbe will form and whole of the cell membrane will pinch out and it will engulf that particle. So that is how what the difference between phagocytosis and the and the endocytosis, phagocytosis and endocytosis. So in that way, then once it enters into the lysosomal fuse, all these things will happen. So that is a difference. So this is about the vesicular we have seen. Now as I told you, we have seen the facilitated diffusion which are channel mediated and carrier mediated. Now what are these channel mediated? The channels, it will have a channels, but there should be some gate to the channels. So these gates are voltage gated, or these are ligand gated. They are voltage gated, or so they are controlled either by the increase or decrease of the voltage, or some ligands or specific things will be there that will control. They are like gatekeepers, and they will control that only that specific substance can enter, other things cannot enter. So now we'll see the details of this in the next class. What are the types of channels, what are the examples involved in that, and what are the disorders that are associated if any of these channels are not functioning properly. That will continue it in the next class.